The talk is about cultured meat production. I'm happy to be here because there's quite a bit of hype created around cultured meat. There's also quite a lot of misunderstandings about cultured meat. There's also some anxiety, as you may have witnessed the recent ban on cultured meat in Italy. So, a very brief history of cultured meat, at least the mainstream version. The idea behind cultured meat is, it dates back maybe to, that's at least the person that's most often quoted, Winston Churchill, who said, well, you know, we produce whole animals, but we eat only part of it. Wouldn't it be great if we could just make the parts that we just eat? Great idea, very well picked up by people that are concerned about animal welfare. So the idea is really to make meat without having to sacrifice animals. So certainly a very worthwhile aim to pursue. Now, that was purely hypothetical. It took until 2013 when a landmark happened, when Mark Post from Maastricht University brought on in a show the world's first cultured meat hamburger. Then it went a little bit silent, not much happened, but then in 2016 there was sort of an explosion on all kinds of startup companies around this idea. So that's the mainstream version. The scientific version of cultured meat is that in the medical world, we've been already studying muscles and how to generate muscles for very, very long, much longer even than I have been living. And I would like to acknowledge one of the pioneers in this field, Herman Vandenberg, who has been studying muscle cultures in the 1970s and then published on that in the 1980s, where he was making what he called bioartificial muscle. So bio because it's based on cells, animal or human cells, artificial because it's still not exactly the same as a muscle in an animal or in a human. And so ironically, because of a lack of human cells, because he was at the medical faculty and he wanted to study muscles for human regeneration purposes, ironically, he only had access to avian cells, chicken cells, and so the first muscles that he made in the 1980s were chicken muscles. So this has been researched quite a bit. There was a lot of interest from NASA. Still is quite a bit of interest from a medical perspective on what we can do with these human muscles. Now, fast forward a bit to 2000, when some research, also sponsored by NASA, was published in a small journal, Acta Astronautica, where they studied fish meat to be produced. It wasn't a huge success, sort of disappeared a little bit in the literature. But then in 2005, there was a commentary in the journal Tissue Engineering, which is really the journal in the field, by which some people said, you know, with all this technology that we're developing in the medical field, why wouldn't we apply this for cultured meat production? And then obviously it took until 2013 until somebody did this. Now I also put there the cover of my thesis in 2005, because what you're seeing there is also some muscles that I made there in the green strips on top of what you're seeing is a mouse, and we made green fluorescent human muscles that we implanted again to study if we could use those muscles for medical purposes. So what happened after this explosion in, say, 2016? Well, depending on how you count them, but a number of hundreds of companies started to evolve in this space. There's approximately about $3 billion that has been invested in the field. And so several of these companies are trying to make poultry, pork, beef, seafood, or related species in the cultured meat space. At this point, as far as I know, only one company has a product that's already on the market, and that's Good Meat. It's on the market in Singapore, and I put it in between brackets on the market because it's only very limited available, from what I understand. One needs a reservation to be able to get access to this product, and reservations are very limited and given mainly to journalists and people that have some influence in the field. So I'll give away the yeast of my story here. If we're talking about cultured meat, I would like to split up two definitions. So in a strict sense, cultured meat would be making biological tissue, often it's muscle that's being used, that we create in the lab or in an industry setting. That's exactly the same as meat. And as you'll see later in the presentation, there are companies that claim that they make something that's exactly the same as the tissue in an animal. Spoiler alert, this is not true. It's not possible at this stage. What we can make is cultured meat in a broad sense. And in a broad sense, what we're doing is we're creating something that has something to do with meat. It's meat-like. We're using animal cells, but we're combining them with other ingredients. And I will now, for the briefness of the talk, call these hybrids. So let's perhaps have a look at what this first hamburger was made of, because it's of interest to know what's going on there in the field. And granted, it's 10 years ago, things have evolved, but not so much as we would like. So how is this being made? On the upper left, what you see is 24 well plates. These are plastic dishes, and in the plastic dishes, little rings are being made. They're made based on animal cells that are connected to each other with an... It's not been defined, so there's not much to find about this first hamburger. That may surprise you, but it's probably fibrin or collagen, an animal product with which the cells are being glued together. So Mark Post made about 10,000 of these little rings, and then they are harvested. You see them in the dish below that. So you see a few hundreds of these little rings. They're like worm-like. We'll go into the structure in those little rings in a minute, but then they're being combined. And then magically happens this first hamburger. Now, the rings don't appear as anything as what you would know to be a hamburger. The color is not right, so the color has to be adjusted by a colorant. Beetroot juice was used. They don't stick very well together, so you need some binder. Breadcrumbs were being used. And then as for taste, it didn't taste by itself very much, so some saffron, some caramel was added, and then it was cooked in some butter. Now, to make this prototype, it costs 250,000 euro. That was sponsored by Sergey Brin of Google. And then what Mark said, and I apologize, it's in Dutch, from a Dutch newspaper. The hamburger from the lab will cost 11 euros within 3 years. Now, if you look at the date, it's maybe a little smallish, but the date says 2016. And so this is sort of the time frame that is being pushed all the time. We're going to make this in 1 to 5 years. Obviously, we're 2023, and there is no hamburger there that I know of at a price of 11 euro. So what is going on? Why are there not many of these products yet? Because there are still a lot of challenges. And time is very limited, so I will not go very deep into those, but I will tackle each of those in the next slides. We've summarized these very well in a paper in Nature Biotechnology 4 years ago already, Challenges in the Quest for Clean Meat. Clean meat is another name for cultured meat or cultivated meat. So first thing is you need cells. So we can take those cells from animals. We don't need to kill the animal. We can expand them. And then we use these as a base for the cultured meat. So there's a number of cell types that are being studied. That's what we do in our lab. We compare different cell types, compare characteristics, and see how well they evolve to muscle. So what you do is you take a few cells, and then the cells are dividing. We can keep them dividing in the lab, but if you want to make the equivalent of a steak or the equivalent of a cow or even more, those cells need to divide quite a bit. So on the y-axis, you see the number of cells that we sort of need to make the amount of meat. On the x-axis, you see the number of times one cell needs to double to reach this. So in order to make sort of the equivalent of one cow, we need to have at least 30 doublings. So a cell needs to double 30 times. And that's very different from the microbial processes that we heard about before, because microbes, they will double in an hour, 2 hours, whereas these eukaryotic cells, they will take approximately 1 day to double. So it means that you need to work very sterile, because if you don't work sterile, the microbes will take over your whole culture. Now, realizing these 30 doublings is already, for a number of cell types, is a challenge. Now, this is sort of where we are at this point. We're able to do these number of doublings with myogenic stem cells. However, in our hands, and also in the hands of other labs in the world, what happens is, if you expand these myogenic stem cells, so the stem cells that are obtained from muscle biopsies, by culturing them, they lose the property to merge and to form myotubes. That's an essential characteristic of making muscle. So we're studying this. I'm positive that we'll be able at some point to overcome these barriers. That's why we're studying that. But at this point, it's still very difficult to expand cells and make these myotubes. Perhaps other cell types, for example, pluripotent stem cells, can be of use, but we still need to do some genetic engineering before they're able to fuse to myogenic-like myotubes. Second problem is, how are we culturing them? Well, we need to feed these cells. We need to give them cell culture media, and until recently, and still a lot used, we used a product called fetal bovine serum, FBS, that is derived from cow fetuses. Obviously, that is a no-go in the cultured meat space. So we're looking for alternatives. The issue with that is that most of the alternatives are really not as good as the FBS itself. So the cells don't divide as long or lose their properties. Again, this is a technological challenge. I'm quite positive we'll be able to overcome that with time, but at this point, it's a huge challenge. And moreover, the cost of these other media exceeds the cost of FBS. So we're talking here about medium costs. Again, this is pharmaceutical products, but we're talking about several hundreds euros per liter. So the cost needs to go down quite a bit in order to become economically feasible. As a side note, we talked about circularity. Well, cows do convert waste streams, products that are not consumable directly by humans. It's really not clear if waste streams can be used for cultured meat, but again, another challenge for this field to look into. Third issue is the bioreactors. So we need to upscale, and we cannot do this the traditional way, that is 2D cell culture. So a lot of people look into how you can upscale these cell cultures, but you need to work in a sterile fashion if you want to avoid antibiotics. It's really not clear to what extent we'll be able to get fully rid of antibiotics. This below image is a screenshot from a CSO of a company that was giving an interview on a documentary, who said, well, we don't use any antibiotics, but in the background there was the laminar airflow with the tube there, where you see the designation 1% P/S, so that's 1% penicillin streptomycin. So probably not many people realize that, but it caught my attention. So then if you can upscale the number of cells that you would need, what you get is an amorphous mass. So a bunch of cells putting together is absolutely not the same as a tissue, right? So we need to have some scaffold materials in order for those cells to grow on, to differentiate on. Also, that is very unsure what can be used. There's been a publication in Nature Foods about textured soy protein on which cells can grow. There's a number of other products that are being studied, but nothing of those really makes for a muscle structure. And so what's being said is, well, we'll use 3D printing. 3D printing is widely cited. However, at this point with 3D printing, one, we cannot fully make a mature muscle, and second, the volume by which we can print is quite limited with the current 3D printing tools. But here comes my main point, and this is something that's hugely overlooked. And so that's tissue maturation. So when we look at the development of a human or of a cow or any other species, what we see is that during development, there's a number of phases that happen. And so for cows, similar to humans, in week 10 to 13 of gestation, what happens is that single cells, the ones that we expand in the cultures, fuse together to form myotubes. That takes another couple of weeks before those myotubes grow a little bit and become myofibers. What the difference is between a myotube and a myofiber is that in the myofiber, there is already the buildup of myofibrils. So these are the subcellular structures, the actomyosin complex that makes sure that the muscle can contract. So this is needed. And what happens during this buildup of this myofibril structure is that the nuclei are being pushed, the cell nuclei are being pushed to the sides, and the whole myofiber fills up with all those proteins that we know, and that then form the structure, the texture of the muscle. So this is sort of what we can do in the lab, what others can do in the lab. What you see is myotube structures with nuclei that are central and that are somewhere in probably the phase where they go at best from myotubes to myofibers. But they're not at all what a muscle looks like after birth. In order to do so, what we need to do is we need to work on technologies where we go from embryonic and fetal structures to have tissues that look more like mature tissues. Now, one problem with that is this phase, going from these fetal structures to more mature structures, costs time, a lot of time, and we probably can speed that up as compared to normal development, but still, time is money. And it's really not clear, and I really haven't seen any good approaches at this point where cultured meat companies are studying these tissue maturation. At best, they already go into the differentiation, going from single cells to myotubes. So that's me and my PhD student there looking at how we can exercise our muscles. We have different ways. I'm not going to go into that. But that's for purely the myotubes. Obviously, a muscle is more than just the myofibers. What we see in a muscle is a very well-structured tissue where there's myofibers that are being surrounded by a very well-organized connective tissue structure. There's also fat in between the muscle. There's blood vessels. There's innervation. I'm not saying that we will need blood vessels and innervation for cultured meat, but just to say that we're still quite a bit away with tissue engineering from what a real muscle looks like. So if you see announcements like these where a certain company says, well, you know, we can make muscle, or we can make meat in a few days, or it is meat, well, that's just a marketing story, really. So then the question is, you know, of these products, what is the nutritional value? What is the safety? What is the sustainability? We have to compare that, obviously, to the farm-raised meat. And farm-raised meat, as you all know, is very nutritious, gives essential minerals, gives products like creatine, carnosine, anserine, and so on, vitamins. Also, the development of muscle to meat, one important aspect there are post-mortem processes. It is not clear at all, I haven't seen any study for cultured meat if there is anything like post-mortem processes happening in these products. Then, if we cannot make something similar to the farm-raised meat, maybe we lack certain compounds. Well, the idea is then, well, we can add that to the final product. So then I think a question is also, if you add it to a final product, do you really need the cells, or can you just consume those products directly? It also begs questions as how do we define meat? So we need to be a bit more specific about these hybrids. And I would also argue that we require novel assessment methods. So all of this has been summarized in a paper by meat technologist Ilse Fraaie in 2020 already, but pretty much we're still at the same level of knowledge as what we had then. So the product on the market then, the company previously was named Just. Now it's Good Meat, so they have in Singapore what they call chicken nuggets. What they're made of, the chicken nuggets, is a culture of fibroblasts. So fibroblasts are connective tissue cells. They're not myogenic cells, so they're not serving muscle really. It's connective tissue at best. And then as for upscaling, as I already touched upon, it's not so easy to do this upscaling. A number of companies worldwide are trying this upscaling. Mosa Meat, the company of Mark Post, recently opened a facility in Maastricht. And so the output of the facility there is going to be a few tens of thousands of cultivated burgers. Now, if you calculate a few tens of thousands of burgers, that should be somehow similar to approximately the meat you get of 25 cows. So that's sort of a small-scale animal farm. And it'd be very interesting to see what inputs go into this facility, what energy will this require, how many people need to work in there, and so on and so forth. So there is still quite a lot of unknowns, and those were summarized in a recent paper in Animal Frontiers. So because of all the misinformation and quite a large lobby of the field, it actually trickles down also to the larger institutions. So this is here a call from the FAO that opened last year. And then the text I highlighted there is sort of copy-paste from company, from what companies will tell you. So products whose composition is comparable or can be comparable to conventional animal products. Methodologies have been well characterized. They're ready to move from lab to production facilities. I would argue that they're not well characterized. In fact, they're very untransparent, and that's opposite to what they claim. So the conclusion here is that still many questions remain, and I would argue that independent academic research is really needed to discriminate facts from wishful thinking and is needed to guide policy. And also that was recently published by Masha Olinik and myself. So with that, I'd like to acknowledge the people working in my lab on both the medical aspects and the cultured meat aspects, specifically Maria Olinik encircled there, who did some of the work that I talked about in the presentation. Also acknowledge the funding I get from the FWO in a project called Custom Meat with colleagues from the University of Ghent. Thanks for your attention.